Well, thank you, everyone, for joining us in person and those of you who are joining us virtually. My name is Bill Gladden. I'm Executive Director of French and Pickering Creeks Conservation Trust, your hosts for tonight. We were established by Sam and Eleanor Morris in 1967, and we've worked with landowners, townships, foundations, municipalities, state and county governments to purchase and monitor conservation easements. And last year we just crossed our 200th easement that we hold. So yeah, exactly. Yes, thank you. Very one of those, like, when you were a kid and you would watch on your road when the odometer would flip. We flipped to 200. It was very exciting. And we have a couple of properties that we own. Two of them I refer to as our signature preserves. And there's a trail map of one on the corner of the table out there, the Thomas P. Bentley Nature Preserve. And we also have another one, Templin Woods Preserve, that is really close by. And we have aspirations to connect the two with a trail. Hopefully before too awful long, but we continue to make trail connections and try and help connect people to the land and the natural resources of Northern Chester County. And one of the ways that we do this is through our lecture series. So thank you very much for coming out tonight. Tonight's lecture is brought to you by a generous sponsor, Virtual Farm Creative. They do marketing, they do advertising, graphic design, copyright, content, and they strategize with websites and developing. Really, whatever you decide to call them, Virtual Farm Cooperative responds with equal measures of focused inspiration and determination to create a perfect personality for your brand. And you can find them at virtualfarm.com. And tonight, we are thrilled to have Philadelphia Mycology Club President Sam Bucciarelli tonight for an introduction to mushrooms and mycology. We'll learn how to pinpoint identifying features of fungi, make high-quality mushroom observations, and gain a better understanding of the role that fungi play in our ecosystems. Sam is a full-time student working as a curatorial assistant in the Herbarium in the Academy of Natural Sciences. She's been independently studying fungi since 2018 and is the current president of the Philadelphia Mycology Club. She especially loves plant pathogens, fungi with highly specialized niches, or otherwise complex ecology, and also anything orange. The Philadelphia Mycology Club is also a 501c3, as is French and Pickering, and we're both nonprofits. Their mission is to study, collect, and document fungi to foster accessibility, diversity, and inclusivity through educating the community on fungi and to steward the land and advocate for fungal conservation. Thank you so much for coming tonight, Sam.
Thank you so much for the warm welcome. I'm happy to be here with all of you tonight. I want to let you know before we start our presentation that we are in the historic collection site of mycologist C.B. Moore. And I am very excited later this year. I'm going to get to take a look at his collection, which lives at the Cornell Herbarium. So I'll be touring the herbarium there in September, and I'm excited to comb through it so that I can compare it to the things that I've been collecting in this area over the last 5 or so years. So that's really exciting for me. The mushrooms of this area are very well documented, especially compared to the rest of North America. I love to start all of my presentations off with this beautiful and colorful slide. A few years ago, I had a work injury that put me on bed rest for several months. And that was when I put together this graphic. But these are all mushrooms and other fungi that can be found within 1 hour of the city of Philadelphia. So these are not tropical, they are not exotic. These are things that we can find right here in our own backyards. So we're going to talk a little bit more about some of these things and the different roles that they play tonight. But first, we'll go over some fungal facts. The first thing I want to tell you is that all mushrooms are safe to touch. In the 1950s and 60s, there was a hideous smear campaign against wild mushrooms. noting that they were dangerous and that people should avoid them at all costs, but that is not true. You can pick up a mushroom, touch it, smell it. You can even taste it if you spit it out, totally safely. Notably, you cannot and should not do that with plants. I'm sure some of you know some poisonous plants in this area, poison ivy being the big one. So all mushrooms are safe to touch. That's the first thing I want to tell you as we embark on this journey together. The second thing is that our fungal diversity outnumbers plant diversity 6-1 in any given ecosystem. So for every one species of plant, there's going to be 6 species of fungi. Notice I did not say mushrooms. We'll get into that a little bit later. So the things that are on the screen right now are 2 phenomena that fungi exhibit. The first one being bioluminescence. So that's what's going on in the top left of the screen. This is Pinellus stypticus. This was photographed in my friend's closet. So this is a mushroom that has been collected, brought back to someone's home, and photographed over long exposure. If we lived in an area that had a lot less light pollution than Greater Philadelphia, we would be able to walk through the forests at night and see these glowing mushrooms as we make our way down the trails. And I have had the pleasure of doing that in the Catskills and in other parts of Appalachia. This is not very unique in the fungal kingdom. There are many species that bioluminesce. And to be clear, the phenomenon of bioluminescence means producing their own light in the darkness. So they're actually throwing off a secondary metabolite, as they call it, a chemical called luciferin, and that's what produces this beautiful green glow. This is not unique to the fungal kingdom, though. You probably know some insects that glow, notably fireflies. And there are also bacteria, diatoms, and other microorganisms that produce a blue light that comes from a different chemical. And then on the bottom left, this is actually a petunia that has had DNA from mushrooms and also fireflies spliced into its genome so that it will also produce luciferin and will glow in the dark. This is currently available on the commercial market. So if you want a glowing pet, you can purchase these petunias. On the right side of the screen, we have fungi and also a plant exhibiting a different phenomenon called biofluorescence, which is when an organism will reflect UV light. So the columbine, the eastern columbine, is reflecting the UV light back to us, and so are these hebuloma at the bottom of the screen. These hebuloma in the daylight are a very boring beige, but under the UV flashlights, they glow this brilliant green. Lots of different mushrooms will fluoresce as well, and so do insects. Both of these phenomenons are thought to be caused by or with the intention to attract nighttime insects, whether that be pollinators or beetles who disperse mycelium. It is thought that these 2 phenomena are caused by the evolution of or coevolution with insects, although the verdict is still out. On this slide, I have 2 organisms that are competing for the title of heavyweight champion, largest organism in the world. In the background, we have the Pando clone, which is a clonal forest of quaking aspens in the state of Colorado that takes up most of the state. All of these trees are coming from one singular root ball. So they are the same organism, even though they are multiple trees. In the foreground, I have Armillaria ostoyae, also known as the humongous fungus. This is a fungal body that is estimated to be over 2,000 years old. It takes up most of the state of Oregon. And so, of course, when I say this mushroom is 2,000 years old and it's taking up the whole state of Oregon, I don't mean that the mushroom is taking up the whole state of Oregon. Which brings me to my next question. What is a mushroom? A mushroom is the fruiting body of a fungus. We could also call it maybe the sporocarp, and it is made to disperse spores. You can kind of think of this as like an apple on a tree. The tree is there for the entire year, and then during flowering and then fruiting season, it will produce an apple. You can pick the apple off of the tree, enjoy the apple, the tree will remain healthy, and you will have new apples next year. So the apple is sort of like the mushroom, and the tree is sort of like the mycelium. And that is a woven web of thin thread-like hyphal cells that create almost like a tissue. In fact, one of the only tissues that a fungus can develop. They will, mycelial mats and hyphal cells will often present themselves as these sort of cobwebby looking structures. If you move a pile of wood, you'll often see them kind of creeping their way along and performing some extracellular digestion, which we'll talk about in a moment.
Are you saying that mushroom spores, I just assumed the mushroom spores found made of other mushroom spores. That happened.
Well, let's talk about it. So the question was, do mushroom spores find each other and mate? So we'll get into that on this slide. Since we've talked about the spores, we can start there. So a spore is almost analogous to a seed, but is very, very, very small. And it can float on air currents. It can attach itself to the body of rodents, on the bottom of your shoe, in a car. Spores are very easily moved around in the environment, which is a very good thing for the fungus. It wants to be moved around to new places and find new spaces to grow. So a spore will land on what we could call a substrate. Usually that's something like the ground in the soil. It might be the body of an insect. It might be the base of a large tree or an old stump. It will find a suitable place for itself, land, and germinate to produce hyphal threads. And remember, those are the cells that fungi produces, hyphae. Hyphae will climb throughout the environment. Remember, performing extracellular digestion. That's when they sort of spit enzymes into the environment, digest what they're about to eat, and then reabsorb the chemicals. And then at some point, they'll encounter another mycelial body, another hyphal thread of a different mycelial body of the same species. They'll intertwine, they'll spend some time together, they'll go on a few dates, and then they'll say, wow, this is going really well. There's been a lot of rain this year. We have this nice oak tree. We should have a baby. And that is when they will merge their cells, enter for mushroom producing fungi. They will enter a state of dikaryosis, so their cells will actually have two nuclei, and then they will produce a mushroom. The mushroom is, in this case, not like the apple is, and it's not a baby, it is the sex. So when you pick up a mushroom, that is active sexual reproduction that is happening in your hand. Crazy stuff. That was a good question, though. So let's talk a little about some ecological roles. The first, and maybe the one that we all think of when we think about mushrooms and other fungi, is saprotrophic. These are our decomposers. The saprotrophic fungi gain their nutrients through decomposing different types of organic matter. Most often, we think about this as being plant matter like leaves or old wood. But I do want you to know that there are saprotrophic fungi that will decompose animal tissues as well. I'll jump over to the right side of the screen. We'll talk about mycorrhizal fungi. Mycorrhizal fungi gain their nutrients through their symbiotic relationships with plants. Most often, we think about this as being woody plants, but like trees and shrubs. But it is estimated that 86% or more of land plants need a mycorrhizal partner at some point in their life cycle. So already we know that fungi are incredibly important for all life on Earth if the plants rely on them. In the center of the screen, and maybe the most exciting thing that's here, unless you're into chanterelles, is parasitic fungus. This is an Acanthomyces species that is growing on a moth. Parasitic fungi, of course, gain their nutrients through infecting and often killing a living host. So if you look very closely, you can actually see the moth's eyeballs in this image. If you have seen the show or played the video game The Last of Us, it's sort of based off of this concept of entomopathogenic fungi. I have chosen to put up an entomopathogenic fungus, but of course, fungi can be pathogens on plants as well. Let's talk a little bit about basic morphology. If you were to send me a picture of a mushroom to identify, which is a very common thing in my life, the first thing I would ask you to do would be to pick it up and turn it over, because the most clear identifying feature is on the bottom, and that's the spore-producing surface. Most often it presents itself in three forms. That's gills, pores, or teeth. The science name for gills is lamellae, so you may also hear that or see that in descriptions if you're looking in a field guide. But they look like the gills of a fish. In the center, we have pores. They're just like the pores on your face. They're like tiny little holes. Sometimes they're also tubes, in the case of a form group called boletes. And then finally, teeth. These are tiny little protrusions that stick out on the bottom, the underside of a mushroom cap. And these are the most common forms of Hymenium that we'll see. So let's talk a little bit about more anatomy. On the top of the mushroom, we have the pileus, sometimes also called the cap. You might know this word or be familiar with it if you are interested in birds. You might be familiar with the pileated woodpecker. They come from the same root. Underneath of the pileus or the cap, we have the hymenium. Again, that's the spore-producing surface. That is where gene recombination is actively taking place. Then we have the stipe. That's the sort of central point of the umbrella shape. You'll notice that I didn't say stem. Stem is a botanical term that implies specialized tissue. Fungi actually do not have specialized tissue. They have their regular hyphal cells, and then they have the cells that produce spores, and that's it. So we like to use the word stipe. You can use stem, though. I'm not coming for you. You may also use the word stalk. That's fine and acceptable as well. At the bottom of the mushroom, sometimes we have a basal bulb. If you're familiar with tulips or hyacinths, you may have planted bulbs. Kind of the same concept. It's just a mass of mycelium at the bottom of the mushroom. Covering that is the universal veil. You can think about this as being sort of like an eggshell that once covered the entire mushroom until it matured and burst out of its universal veil. The universal veil is also called a vulva. That's vulva with an O in the literature, so you may read that. And then finally, we have the partial veil, which is covering the hymenium or the fertile surface as that space is maturing to protect it from bugs and keep it moist and prevent it from drying out before it matures. So these are all features that you would want to pay close attention to if you're looking at learning to identify mushrooms in the wild or in the lab. So now I want to talk to you a little bit about fungal taxonomy. So we'll start off really big with all life, and that includes 3 domains, which is bacteria and archaea, and then eukaryotes, which would be us, and also the fungi, plants, protists, and some other stuff. We'll talk a little bit about that later. Within eukarya, we have the kingdoms. We have plants, animals, fungi, protists, and again, maybe some other stuff. Taxonomy is sort of always in flux. Beneath kingdom, we have phylum, which is what I'm going to talk to you about today. There are 2 main mushroom-producing phyla within the kingdom of fungi. The first of which is Basidiomycota. Basidiomycota, or basidiomycetes, as you'll often hear them called, are what you would typically think about when you're thinking about mushrooms. That's like the Mario mushroom, the fly agaric, the button mushroom, the typical mushroom shape with the cap and the stipe, and the gills underneath. That's what you would typically think of. That's Basidiomycota. One way that I like to tell people that you can distinguish this is that Basidiomycota has its spore-producing surface facing downward towards the basement. They passively release their spores using gravity. In this image, we have a cross-section of the gills, and then we also have the basidium, which is what this phylum is named for. That is the specialized tissue that they have to produce spores. Most mushrooms have four-spored basidium. You can get into that if you get really deeply into mycology, but most of the time, fertile surface is facing down. It's a basidiomycete. Here are some examples. We have the coral mushrooms that produce their spores at the tips of their coral fronds, bird's nest fungi in the center. These are also called splash cups. So they will wait for rain to break open these little tiny spore packets that look like eggs and disperse their spores due to the force of the drop of rain. On the far right, we have stinkhorns. This one's actually called stinky squid is the common name. And this one produces a sticky and smelly substance called gleba that carries their spores and is very, very attractive to flies and other insects. So the flies will come and land on the gleba, pick up the spores, and carry them off to new locations. On the far left, we have puffballs. Similar to the splash cups, they kind of wait for rain or for something to step on them to release their spores into the air. Jellies on the top right. This one in particular is called witch's butter. It produces its spores within its folds. And then at the bottom, we have a resupinate crust. Resupinate meaning that it's facing outward and flat on a log. And then finally, in the basidiomycetes, this category, rust fungi, is what I specialize in. If I had a specialty, it would be these. Rust fungi are extremely interesting. I could talk to you for multiple hours just about rusts. But what I think is really interesting about them is that many of them cycle between two different plant hosts. So they're obligate plant pathogens that grow on living tissue. And like the one in the center here, the cedar apple rust, it will spend several stages of its life on an apple tree and several stages on a juniper tree, pictured here on juniper. So they are incredibly interesting. They have these interesting ecological niches, and of course they're orange, my favorite. Next, I want to talk to you about the phylum Ascomycota. So this is the other phylum that produces mushrooms. Ascomycetes have their spore surface facing upward towards the attic. Their fertile surface comes in a few different forms. Don't feel the need to memorize these, but typically it's just about how open or closed that hymeneum would be. Here are some examples. Truffles, that's a big one. This one is a closed structure that grows underground and smells bad, depending on who you ask, with the intention of attracting mammals to dig them up and disperse the spores. On the bottom left, we have the hard, or as I like to call them, crunchy ascomycetes. Ascomycetes actively use energy, usually, to release their spores. So when you find these strange, crunchy, hard structures, know that they are working very hard to push their spores out. Did you have another question? No, okay. In the top right, we have cups. These are called blue elf cups. These typically will wait for a gust of wind to come by and then will, again, use energy to release their spores as that gust of wind has been detected in the hopes that it will allow the spores to be picked up by the wind and carried off to a new location. Morels on the bottom right are essentially a bunch of cups on a stipe. Let's talk a little bit about lichens. Lichens, taxonomically, are fungi, and they are mostly in the phylum Ascomycota. Emotionally, most people who are studying mushrooms are not interested in lichens. We kind of leave these for the plant people. The plant people leave them for us. The lichen people are a breed of their own. But they are fungi, and I want to talk to you about them tonight. Lichens are a whole little ecosystem. They're usually 4 or more components: an ascomycete fungus, an algae, a cyanobacteria, and a basidiomycete yeast living within the structure of the ascomycete's fungal body. In the terms of how this evolved, you can think about it like this. You call and order a pizza. You say, "Hi, can I have one plain cheese pizza, please?" And the delivery man comes to your house with the pizza, and you say, "Hey, do you want to come inside and eat this pizza with me?" And he says yes. And you eat the pizza together, and then he moves into your house and makes you pizza every day forever and ever for the rest of your lives for millions and millions of years. And that is what happened with the lichens. An ascomycete fungus engulfed an algae and a cyanobacteria with the intention of eating them. They continue to photosynthesize within the mycobiont, and they decided it was working out. So they continued to work together and to live together for millions of years. Lichens are indicators of good air quality. So if you see lots of lichen diversity, you can pretty much guarantee that the air is clean wherever you're located. And they are not damaging to trees. So if you see them on your trees, you don't have to be concerned. Because they are photosynthetic organisms, they like to be perched high up where they can get more sun, especially in the winter months. And so they are not harming the tree. They're just kind of resting on it, much like the mosses do. Yes.
So in my yard, I have lots of trees. And the silver maple tree, every time a branch falls, coated with lichens. Sycamore tree, you don't see any lichens. A lot of trees, you don't see any lichens on the branch, and then particularly silver maples if they're photosynthetic, why would they care what tree they're on?
That's a great question. So the question for my friends online was, why would lichens choose one species of tree over another, particularly asking about a silver maple versus a sycamore? And in this case, the answer is likely related to the textures of the bark. So the sycamore exfoliates its bark. It's rather smooth, doesn't have a lot for a lichen to attach to. But the silver maples, although they are rather flaky in their bark on the trunk, their branches have texture, but they don't exfoliate their bark. And so the lichens have a lot of texture to grab onto. Like if you were climbing up a stone wall versus a brick wall, it'd be much easier to attach to the texture of the stone. Excellent question. So we have a lot of lichen diversity in this area. Lichens were almost killed off due to the chemicals being produced by leaded gas. And then when the switch was made to unleaded gas, we started to see a return of lichen diversity in urbanized areas. And so there's 50 or so common lichens in urban areas in what is called the Mid-Atlantic megalopolis. And it's very easy to learn all of the lichens that grow in the urban areas. So I would encourage all of you to do that if you're interested in lichens. Next, I want to talk to you about slime molds. So slime molds are not fungi taxonomically. But they are studied by mycologists because they grow in a lot of the same places as mushrooms, like wet, well-decayed, decomposing logs where we're already poking around. They come in all sorts of bright colors and fun shapes. They're very usually, but not always, very small. And so they are often studied by mycologists, despite the fact that they are in a whole different kingdom of life. Slime molds are in the kingdom Protista, which is a junk drawer for anything that did not fit nicely into the other three kingdoms. And it also includes brown algae and like brain-eating amoebas and like lots of things are in the kingdom Protista. That needs tons of work. If any of you are looking for a project for your PhD, consider slime molds or other protists. So we have some slime mold diversity here on this slide. In the top left, we've got wolf's milk slime mold. I really love these. If you can pop them like log pimples. It's gross, but I do encourage you to do it. It's a fun way to engage. On the top right, we have the pretzel slime. Then we have the Ceratio mixa in the middle, sort of has this honeycomb structure to it. On the bottom right is cotton candy slime mold, the middle chocolate tube slime, and the yellow one is dog vomit slime. I know. If you are walking around outside when it has been rather humid or particularly wet, you're very likely to find slime molds. They like to be in moist areas. It's also worth noting that all of these structures are spore-producing structures, and these also live as microorganisms outside of our realm of perception, much like fungi do for a lot of their life cycle. And that is also one of the appeals for being studied by mycologists. There is something psychologically that most people who choose to study mushrooms are chasing this ephemeral thing that appears and disappears in short amounts of time. That's why we don't like lichens. The lichens are around all year. They grow over the course of many years. And what you see is what you get with the lichens. This is its whole body versus a slime mold, which kind of disappears. Very fun. So I want to talk to you a little bit about some common edible mushrooms. But first, I want to go over some best practices if you're interested in foraging for mushrooms, or if you're interested in collecting for science, which I'll get into after the edible section. The very first thing that I want to tell you is that if you are going to eat mushrooms, the thing that will save your life if things go wrong is that you took a picture of the exact mushroom that you ate. This is critically important. But as we'll talk about later, there aren't very many toxic mushrooms in our area. Please take pictures of what you're eating. I also want to tell you that foraging for mushrooms and other wild edible foods varies a lot place to place on whether or not it is permitted. So in Pennsylvania, it is permitted to collect mushrooms, seeds, and berries from nuts from any of the state parks. So nearby to here, we have French Creek State Park and also Marsh Creek State Park. Foraging is permitted in those locations. However, I live in Philadelphia. Collecting any wild growing thing for any reason is not permitted within the city limits of Philadelphia on any public lands. And these are very important things for us to know. There are lots of reasons why these laws might be in place. It might be that, particularly in the case of Philadelphia, it may not be safe to eat things that are growing wild in heavily industrialized areas. The other reason has to do with conservation. So mushroom foraging is generally considered to be a sustainable activity. Like I mentioned earlier, it's very much like picking an apple off the tree. It doesn't harm the tree to pick the apple. However, not everything that we do while foraging is considered to be sustainable. If we decide that we're going to walk off trail and trample through the leaves over the spring ephemeral wildflowers and squash the habitat for the baby salamanders, that is an unsustainable practice. And so I want to encourage anyone who is interested in foraging or collecting for other reasons to be mindful of the habitats in which these fungi are growing and the other things that may be sensitive to our presence. Let's get into it. First, we have chicken of the woods. This is a very easy to identify mushroom. It is orange and yellow and very large. I typically don't find it smaller than a dinner plate unless it's very immature, but I very often will find it larger than a dinner plate. Like on a large log that has fallen over, you might be looking at like an entire sofa worth of chicken of the woods. So this is one that can be extremely prolific, and it also fruits pretty much all through the growing season. I found it as early as March and as late as November in good, fresh, edible conditions. This is a great one to learn. The underside is yellow and has pores. There is another species of Laetoporus that is white pores, but they are both wood decomposers and fairly easy to identify. Nothing that looks like this is considered to be toxic in this part of the world. You will not find it with pines, but you can find this with pretty much any other woods, maples, oaks, tulips, beech. I would say I find it most often with oak and tulip. Next, I have oyster mushrooms. There are a few species of oyster mushrooms that grow in our area. Pictured here, we have Pleurotus ostriatus. This is a species that will grow all through the year. And I'm talking like January, it's got snow on top of it, and it looks fresh and beautiful and ready to eat. This is recognizable because, again, it is a wood decomposer. It is growing directly on a standing or fallen piece of wood. The gills go down the stipe. We call that decurrent. And the caps are beige to white in this species. The summer oysters are all white, and we also have the invasive golden oyster here, which is yellow. Next, we have morels, everybody's favorite in the springtime. People get really excited about morel season, which is very short. It runs from about mid-April to mid-May, sometimes even less time than that. Morels have an interesting ecology because they are considered to be mycorrhizal, meaning that they have an association with a tree host, usually cherry, apple, ash, elm, or tulip. However, once their tree host dies, morels will pivot their ecological role and become the very first decomposer to start breaking down the woody tissue. This is very much like the microorganisms that live in our bodies. We have tons of microorganisms in our digestive tract that are helpful to us while we're living and eating food. And then as soon as we pass on, they are the first things that will begin decomposition. So morels are very similar in that way. Next, we have chanterelles. This is the most popular edible that's out right now. This is also a mycorrhizal species, so it will grow directly out of the ground. This is important for this species. You always want to make sure you're observing the ecological role. Are we growing out of the ground? Are we growing in wood? Are we growing on a dead bug? What is the ecological role? That's very helpful in identification. Morphological features that would help you with identification of chanterelles. It has these lovely forked gills. You can see that they're very irregular. They sort of crisscross each other, and they're also very shallow. You will sometimes hear that called false gills. We could just call them gills. That's okay. There are also smooth chanterelles in our area, which look very much like this, except they're smooth instead of having these shallow gills. All species of Canthrellus that grow in this part of the world are considered to be edible. We also have tiny little orange chanterelles that were pictured earlier in this slideshow that are common this time of year as well. Next, we have lion's mane. This is a plant pathogen. So this one will often be found growing on a tree where it has lost a limb in a storm or due to other human activities. This will move into that space, begin infecting the tree, and will actually fruit right out of that wound. This will also pivot to become a decomposer after the tree has died. This can kill a tree. I will say of the lion's mane, it is a native species to this region. And so although it does tend to kill its plant host, it's not something that I'm ecologically concerned about from a land management perspective. If you have a tree in your yard near your house and it begins to get lion's mane on it, consider that it might be time to call your local arborists. But it's not something that I worry about being present in the forests. Question, I have found this most often on oaks and beeches, but I have also found it on maple, maybe just maple outside of oak and beech. Next, we have Old Man of the Woods. This was the first mushroom that I ever tried to eat, or first wild mushroom that I ever tried to eat. It is very woolly in texture. I think Old Man of the Woods is a really good name for it. It sort of gives like, it's giving like Bernie Sanders with his mittens, you know? It's a great name for this mushroom. When it's young and fresh, it has these beautiful striking white pores. With age, they stain to black. And actually, when you cut this mushroom open, it will oxidize and stain a very bright red. Like think the color of raw beef. That is the color that this will stain. It's very impressive if you can find a fresh specimen. I did not enjoy this as an edible. But as I found out later, I actually don't enjoy any mushrooms to eat. So although I am a local expert, I am actually reaching into the realm of academic mycology. I am not interested in eating any of them. Which is good. That means if you hike with me and we find some good stuff, you get to go home with you. Next, we have black trumpets. These are related to chanterelles. They're in the genus Curatorialis. They're hollow all the way through, much like a little bugle. Although they're called black trumpets, they come in many different shades, black, gray, beige, even reaching into oranges and yellows, like their cousins, the chanterelles. These are not uncommon in our area, but they are very hard to see because they blend in well with the leaf litter. I'm going to give you guys a pro tip. I always find these in erosion pathways where I have seen that leaf litter has moved due to rain or gravity or other forces. That's where I will see black trumpets pop up. These are mycorrhizal, usually with oak and beech. Hen of the Woods, this is another favorite among people who are interested in consuming wild mushrooms for the same reason that morels are. They have a very short season, usually running from late September to around the end of October. It's hard to see the scale of this picture, but they are, roughly the same size as a dinner plate in all dimensions, if a dinner plate was like spherical. And the Latin name for this is Grifola frondosa, because it does have these very delicate fronds. On the underside of the fronds, you'll find small white pores. These are mild parasites on well-established oak trees. You maybe could guess that from the picture here. We've got the oak leaf, the acorns, and this sort of crook in the root flares where the hen of the woods likes to pop up. This one's also called maitake. Shameless plug. Every year, the Philadelphia Mycology Club hosts a Halloween party and jerky contest using maitake mushrooms to make jerky. Despite the fact that I don't eat any mushrooms, I was the 5-year champion, and I have since been replaced, thank goodness. Next is the giant puffball. This is a grass decomposer. You'll find these in grassy fields and roadsides. It looks like a soccer ball. There isn't really another way to describe this mushroom. It's about the size of a soccer ball in this region as well. In the Midwest, you may find them like this big. They're very large. But around here, we'll see them get to be about the size of a soccer ball before they mature and begin to produce spores. It's very important when you find what you think is a giant puffball to cut it in half all the way through and make sure that it is white and a consistent marshmallowy texture all the way through. In my mind, it is hard to confuse this with a deadly amanita, but poisonings have happened and it has been confused. So what you need to look for is a consistent marshmallowy texture all the way through, all white.
What's the color? All white.
So there are earth balls that are smaller. Sometimes they get to be this big, maybe hamburger size. And when you cut those open, they're purple on the inside. Also, this one, when it matures, the spores are like an olive green, and it's just gross. It's not good to eat at that point. So if it's not white, don't eat it, even if it is this species. Next, we have resinous polypore. This is sort of the last of the fall mushrooms before we move into the winter stuff. This is a shelf mushroom. So it can grow on standing dead trees or fallen trees. The view of this image is sort of like looking down at a fallen tree. So this is the cap, the top of the mushroom. On the bottom, we have a pure white pore surface, which you can see along the edges. The edges of this mushroom are incredibly tender and juicy. This is one of the ones I would eat if I was going to eat them. It makes a good bacon substitute, but you want to harvest just the edges, the tender parts. Lichens are usually shades of green or yellow. There are lichens that grow things kind of like shelves, and there are lichens that will grow on shelf mushrooms. The term shelf mushroom is like a form group, so it's as much an identification as tree. Another thing about this particular species is that it often gets these guttation drops, which you can see on the left side of the screen on the cap. These guttation drops are sort of sticky liquid that will help to spread spores around. And that's very common with the resinous polypore and how it got its common name. So I want to talk a little bit about toxic mushrooms. We don't have very many in our area. Did you have a question? So the question is it better to cut a mushroom or to pick or pluck a mushroom? And the answer is whatever is working for you. I personally, if I was going to forage, would prefer to cut them only because it prevents dirt from getting into my basket and all throughout my mushrooms. There is no ecological reason to cut versus pluck mushrooms. So if you're not interested in getting like down onto the ground to cut them, you can pluck them and know that you're not doing a disservice to the environment. That's a good question. So moving on to toxic mushrooms, we have a few species in this area that I would like to talk to you about. On the far left, this very tall, slender, purely white mushroom is Amanita section phalloideae, which includes the destroying angel and, oh, what's the name? The common name for the other one? Death cap, yes. So that is the most deadly section of Amanita. And I would give you the advice as somebody who is new to foraging that you Avoid eating anything that is tall, slender, and white until you have gained knowledge of some of the nuance of the features of different mushrooms. The things that I've shown you in this presentation are widely considered to be safe, to not have any lookalikes that are toxic. And so there are some things that might be considered to be edible that look like this. But generally speaking, it's wise to stay away from amanitas if you're new to foraging. So amanitas contain a liver toxin called amanitin that has to pass all the way through your digestive system to cause you harm. So you can pick this mushroom up, you can photograph it, you could, and I don't recommend this, you could go as far as taking a bite, chewing it up, and spitting it out, and you will be safe. I don't recommend doing that, but it is this, what's called the spit test, is a common test for distinguishing features between several different species in some cases. On the far right of the screen, we have Gallerina marginata, sometimes called funeral bells or the deadly Gallerina. These contain the same toxin as the amanita, that's amanitin. And they are deadly toxic. If you eat either of these species, the symptoms are that you will find yourself to have had a delicious meal. Several hours later, you'll become ill, then you'll get better. And then within a week, all of your organ systems will go into organ failure and you will die. There is no cure. Their treatments are symptom supportive, so it's not like an automatic death sentence, but you will be immediately put on a liver transplant list, and it is a very serious condition. So I would avoid eating tall, slender things and things that are small and brown. The reason that I put the Gallerina on here, and I do have to mention this, is that the Gallerina look remarkably similar and grow in the same habitats as our local psychoactive species. And so this is one that I feel concerns about people accidentally foraging, because they are small and brown. Most people who are interested in edibles would pass them by. You'd have to harvest so many of them to have dinner. But if you mistake them for something else, it could become a deadly situation very quickly. So I will tell you tonight, and I do not condone unlawful behaviors, but the way that you distinguish these two mushrooms from each other is that the deadly Gallerina will have a rusty brown spore print, and Psilocybe ovoidiocystidiata will have a purple spore print. If you're in a situation where you need to know those things, I want you to know them. I want you all to be safe. So these two that I've talked about are deadly poisonous, but the two that are in the middle are not so deadly. Mushrooms produce a wide range of chemicals. Earlier in this talk, we talked about luciferin. We also just talked about psilocybin, amanitin. The orange mushroom here, the jack-o'-lantern, Omphaliotis illudens, produces a chemical called illudin that will cause major gastrointestinal upset. So this one will not kill you, but you will become very ill. Will not kill you, probably, but you will become very ill if you accidentally eat it. Some people do confuse this mushroom with chanterelles. Although I think that they are very distinct. One of the ways in which you can tell them apart is that jack-o'-lanterns always grow on wood and chanterelles always grow out of the ground. So those are the distinguishing features. And one of the reasons why it's important to notice the habitat in which the mushroom was growing. Finally, these two little deep red, almost black mushrooms are Inocibe taquamenensis, or as I like to call them, taco men. And these are not toxic to people, but they are deadly toxic to dogs. And so if you have a dog that you hike with and that you care very much about, it's important to be careful about what the dog might be picking up and consuming while in the woods. So the compound that is here in this Inacibe is muscimol, which binds to GABA receptors. And if a human consumed it, it would make you feel ill and kind of drunk, but in a bad way. But it would be deadly toxic to a dog. And so these are some of the wide range of toxins that can be present in mushrooms and some of the common mushrooms in our area that are considered to be toxic. If you are considering foraging mushrooms, or if you are looking to observe fungi as you walk around in the woods, here are some ways that you can take photos that will provide helpful information about identification. The first photo on the top left is an in situ photo. That's before you've even touched the mushroom, so we can get a little bit of an idea of where this mushroom is growing. So we see, in this case, this jack-o'-lantern mushroom is growing in a parking lot on a stump. We get a little bit closer, we get a picture of the mushroom cap. We see that it's smooth and orange, and we get a little bit of a view of the growth pattern. This is growing in a cluster. The next picture we need is the spore surface. That's the hymenium, the gills, pores, or teeth after you've turned the mushroom over. These are the 3 pictures that I want you to take every single time. But then moving on, there are some other observations that I'd like you to make for certain genera as you enter your mycological journey. Sometimes it's essential to dig up a mushroom and see that basal bulb and the vulva, the universal veil that may be covering it. Particularly important in identifying mushrooms in the genus Amanita. You also want to notice details about the presence or absence of that partial veil. In this beautiful image, we have Amanita amirarubescens with this very 90-degree parallel tutu that it's wearing. Next, we have the spore color. So as I mentioned before, Seleucidae have a dark purple spore print, and Galerina has a rusty brown spore print. So spore print can be a helpful indicator of what you might be looking at. In this image, we have a spore print that has been left behind from an overlapping cap. That can often be helpful. They will sometimes leave spore prints in the grass or on the logs that they were growing on. And you can often denote the color from just what the gills generally look like. That's a skill that you may develop with time. Yes? So the question was, our friend from the audience had a mushroom come up in her yard and had her work that was long and finger-like, and it was the color red. So this was a stinkhorn, probably in the genus Phallus. Yes, you heard that correctly. Mushrooms, or mycologists, tend to be pretty direct with what things generally look like. And so I would venture that that's what you were looking at. So most mushrooms in the genus Phallus, as far as I know, are considered to be edible, but they're like goopy and they smell bad. So if you're an adventurous eater, if you're like, yeah, I would totally try escargot, give it a try with the stinkhorns. That is not for me. But more power to you if you find them and you're interested. They're most often consumed in their like egg form before they hatch into this finger-like protrusion that is the mushroom. Yeah, no problem. So a few more things about making observations. Like I mentioned with Old Man of the Woods, it oxidizes after you've cut it in half. Some mushrooms will stain blue, some black, some green or purple or red. So for certain taxa, especially, or certain form groups like boletes or the genus Lactarius, cutting them in half can be a helpful identifying feature to see where it oxidizes. And then finally, some general information about your geographic location. I don't expect you to tell me that you found morels behind the Washington House in historic Yellow Springs, but you could tell me I found these morels in southeastern Pennsylvania, and that's good enough. But it can be helpful for identifying two species in different regions. The best way to learn how to identify mushrooms, though, is to join a mushroom club, which is what we're doing here tonight. These are images from forays that my club has hosted. So pictured on the right side is my friend Beth standing around the identification table. At a foray, we go out as a group, collect all the mushrooms we can find, bring them back to a central location, put identification labels on them, and talk about their features in real time to identify them and help you to learn how to identify them yourselves. We also keep track of all of the species that we collect and provide lists to the state of Pennsylvania and all of the park management so they know what's growing in their parks. We also have tons of resources. That's what's pictured on the left. These are all the textbooks that I drag out of my house to share with all of you at the forays. And then I want to talk to you about becoming a community scientist. So what I was just talking about was an event called a foray, in which we keep a species list. We also take photos of everything that came back to the table because we are participating in community science. I am a full-time student and I've just breached into the realm of academia and being a professional mycologist. But for the last 7 years, I've been a community mycologist. I'm learning and studying and providing information and performing population studies as really just a girl. I use an app called iNaturalist that I want to encourage you to use as well. If you don't have a smartphone or are averse to using one, they also have a website, inaturalist.org, where you can post photos from a camera. And it's going to use machine learning to try and tell you what it is that you're looking at. So you might post this photo of Mycena leana, and it will say, yeah, we think that's in the genus Mycena, or it might know right away. It's getting better and better with more photos being added to it. But then some nerd like me can come along and say, yes, that's totally Mycena leana, or that's totally chicken of the woods. And you can know that what you found was something cool or something edible or something exciting. The best thing about the data from iNaturalist is that it can be used by scientists, conservationists, and land managers all over the world who are excited and interested in the things that we see on the trails every day. This is particularly important in the field of mycology. There are 200 maybe professional and academic mycologists in all of North America. That is an extremely low number compared to botanists or animal biologists. But the culture is changing. In 2023, the state of Pennsylvania hired a conservation mycologist to join the Natural Heritage Program. And in 2020, I was part of the rebrand of Fungal Diversity Survey, who puts together these Northeast or these rare fungi challenges. This is the one for the Northeast region. And this is basically a scavenger hunt to say, we think these things are rare. We've put together all this information about their habitats, where they've been found growing, and we want you to see if you can find them because nobody has seen them. And what's really cool about that is that sometimes we do find them. So on Sunday, this past Sunday, my club had a foray at French Creek State Park. And the foray was going particularly well, especially for how dry it's been. When we got back to the table, my dear friend, Paul Stanley, brings me over this dreadful specimen and says, I found this really weird thing. I think it's a truffle. And I looked at it and I knew right away, this is Wynea sparasoides. The last record of Wynea sparasoides, actually the only other record in the state of Pennsylvania, was in 2004. So it has been absent from the records for over 20 years. And we found it in French Creek. This is a pretty big deal. I was on the phone with our state mycologist, Hannah, earlier today, talking to her about the experience of finding it and what she thinks the conservation plan should be for this area. So when I get home tonight, I'm going to send an email over to the park leaders at French Creek and say, I found this really exciting species. We're collecting it for DNA. Next time we find it, we're going to leave it behind because we want it to be able to spread its spores. We're going to photograph it. We're going to be really excited. But we want it to continue to grow. And one of the reasons for that is that Hannah seems to think, and there are scientific papers that are decades old now, that seem to think that this species has a special relationship with the rodents in the area. And Hannah specifically mentioned the redback vole, which is not pictured here. A little harder to find a good photo of a vole. But it is interesting to me that we've not only found this rare fungus, but now we're maybe now that we have an idea of a place where it grows, we can perform a more longitudinal study about the ways that it's interacting in the environment and be able to advise in which areas might be worth prioritizing as high conservation within the parks. And that's really awesome. So you already heard a little bit about the mission of the Philadelphia Mycology Club. The IUCN is the organization that puts things on the red list. The last time I checked, there had only been 800 fungal species assessed worldwide by the IUCN for whether they need a conservation status, as compared to 70,000 plants and over 60,000 vertebrate animals, which is really crazy to think about because earlier I told you that fungal species outnumber plant species 6-1 in any given ecosystem. And so it is really important that we continue to push for conservation and push to get data and push to get these species protected because it will help us to protect the land in which they are growing. Luckily for us, French Creek is not in any danger yet. So one of the things that we do within the city limits as a club is that we have monthly trash cleanups. So these numbers need some updating. These are from 2023, but in 2023, we collected 3,086 pounds of trash out of Cobb's Creek. We also have a native plant garden that we tend to. We put in 400 hours of volunteer work just in land stewardship. We also rescued 5 abandoned animals in 2023. So I want to provide you with some resources here. We have my favorite mushroom field guide. The cover of this book has changed, but the book is called Mushrooms of the Northeastern United States and Eastern Canada. The author is Tim Baroni. This is a very comprehensive field guide that does fit in the biggest pockets of my cargo pants. So it's easy to carry around. It's organized by spore color, and it has a broad focus. So we can learn about ecology, we can learn a little bit about diversity, and it's not focused just on edibles. And then I also have this QR code, which leads to my website, phillymycoclub.com/resources, where I've compiled tons of resources within the area and online resources as well. And finally, I would love to ask you to join the Philadelphia Mycology Club. It is as easy as signing up for our mailing list. You would do that on our website, phillymycoclub.com. All of the events that we put on as a club are free. We partner with a lot of other organizations to bring fungal programming to their communities, and we do a lot of really good work, I think. I also want to tell you before I wrap for questions that we do have a really big event coming up. It's our 2nd annual Fungadelphia Festival. So it is a science and arts festival. We have 2 academic mycologists coming in to speak. I have an industry mycologist who will be running a mold and microscopy table. We're going to do natural dyeing of wooden mushrooms. I have a band coming. And there's going to be free food plus vendors. It's going to be so much fun, and you're all invited.
When?
Oh, August 23rd. It's at the Schuylkill Center for Environmental Education. With that, I am ready for questions from the audience and then from the chat.
Right. Looks like something.
Yes. Yes, I do. So
Question is, what is a fairy ring?
the question is, what is a fairy ring? And a fairy ring is when a mycelial body has expanded from a central point to form a circle, and then because fungi are heterotrophs, they need oxygen, so it'll be exposed to oxygen and will produce its fruiting bodies in the circle around the edge of its mycelial body.
Is there a certain type of mushroom that creates that?
There are mushrooms that are more prone to doing it than others, but there are a number of species that will do it. In this area, I most commonly see the green parasol mushroom, also called the vomiter, forming fairy rings. And they're large, tall, white mushrooms that are very delicate and beautiful, but also will make your tummy very upset if you eat them.
Oh, sorry.
That cedar apple rust on it.
Yeah. So the question is, do I have any tips for dealing with cedar apple rust on a service berry? So the first thing I will tell you, because I love rusts, is that there are many species of gymnosporangium. No telling which one is on your service berry. But my advice for managing the infection that your tree has is to tend to your tree regularly. And by that, I mean to water it. Typically, in years where we have a very wet spring, we see more service berries without rust on them. So, and that's kind of unusual in the fungal world because mushrooms are essentially water balloons. They need water to come into existence, into our realm of perception. But because this is a plant pathogen, it's taking advantage of the plant's poor health. So you'll see it more often if it's been dry. So I'd recommend watering your tree very regularly throughout the late winter and into the fruiting season, and that should help. I never recommend fungicides except in extremely severe cases and if we're dealing with invasive species that may spread.
So from the virtual audience, we have a question: when looking for mushrooms to consume, how close to a road is too close? And too close to an area where pesticides or herbicides are used? I heard mushrooms can pick up toxins from the surrounding area. Is that true?
That's a good question. Yes, that is true. Mushrooms absolutely can bioaccumulate toxins from the environment, and that is definitely an area of concern. I would say use your judgment. Think about how often is this road being traveled? Are you on a gravel road? Are you on a county road? Or are you on I-76? Because that will tell you a little bit about how much this area has been exposed to different types of toxins. And then I definitely recommend reading environmental cues. So you can usually tell if an area has been sprayed with herbicides, definitely, because there will be extreme dieback among the plants. And you can even sort of, if you pay very close attention, you can even sort of see that year over year after application based off of what will come back in the area. And so that is really about being in tune with the environment, not just learning to forage, but learning about different types of habitats and nature's cues, which I definitely recommend. And I do have some resources about that if you're interested. There's a great book series. It's by John Eastman. It starts with forest and thicket, but he covers a few different habitats. And he talks about the different things that associate with certain plant species. And then another really good book by Tom Wessels called Forest Forensics. That's a really good book as well about reading the cues of the landscape and knowing a little bit about the history of what has happened there. From the audience.
Hi.
They are beautiful. Just like lesser celandine is beautiful. So the question is, can I tell the audience a little bit about the invasive mushrooms in the area? So we are talking about the golden oyster mushroom. It is yellow on top. It has delicate white decurrent gills. They grow in clusters on decaying wood. They were released from captivity and cultivation by people who were cultivating them because they are delicious edibles. So I hear. And they have become pervasive in our forests. They are absolutely outcompeting our native species, particularly the summer oysters, Pleurotus pulmonarius, and they are reducing the available habitat for our native species. There isn't really a lot of information out there about that because these fungi do live as microorganisms for most of their life until they produce a mushroom. So they're generally hard to study. But what I can tell you is that you should not buy cultivation kits for golden oysters. If you do encounter them and you want to eat them, right on, but you should bag them in plastic so that they don't spread spores around. If you find them in poor condition where they are no longer edible, I would recommend bagging them up and putting them in a trash can to try and control the spread of spores in the environment. I will say that golden oysters are not the only invasive mushroom that we have. We have tons of invasive fungi. Think about how many invasive plants we have. We definitely have probably as many, if not more, mushrooms and other fungi that are invasive in the area. There aren't really good ways to get rid of fungi in the environment, aside from like widespread fungicide use, which I don't recommend. And so what we're looking at now is like, can the species be naturalized? Are they causing major damage in the environment? And can we control the spread? So those are lots of things to think about as we move forward with sustainable practices, making good choices, growing native species, and learning and connecting with each other and with the environment. Any other questions? Yes. Yes, we do. So the question was, do we have the mushroom that has psilocybin in it? And we have many, actually. They are, like I said, that mushrooms produce tons of chemicals as secondary metabolites, just like our bodies do. And there are many different species that produce psilocybin, hundreds of them around the world. There are probably 10 in this area. There are 3 in the genus Psilocybe that will grow in the state of Pennsylvania, and a few others that are commonly harvested by people who are interested in that particular aspect of mycology. Yeah, I mean, it's a good question. It's definitely a talking point for sure. Like I said, I don't condone unlawful activities. I want everyone to be safe. I want you to be engaged in mycology. And I want the studies that are going on at Penn and Johns Hopkins to continue, because I do think it is promising for medical uses. But I think that it's not the right choice for everybody, and there needs to be more science done before it gets released to the world. Yes.
I read something once about how mushrooms are being used to clean up like toxic waste sites and superfund sites, things like that. You allow mushrooms to grow, they take all those toxins out of the soil and you harvest the mushrooms and put them somewhere else. Like, have you seen much about that?
So the question is about mycoremediation and the potential for mushrooms to accumulate and remove toxins from the environment. Yes, a short answer, yes, they can do that. There is a guy named Peter McCoy who used to or maybe still does run the Oregon Mycological Society, who trained oyster mushrooms to grow on and decompose cigarette butts, which was a huge deal, like used ones with nicotine in them. That was a huge deal and is very promising. In December, I will have someone speaking to my club about mycoremediation in Crum Creek using turkey tail. It was a study that they did during their time at Swarthmore. I'm really excited about that. This isn't a topic that I know a whole lot about, but it's something I'm really interested in. I'm more on the biodiversity side of conservation than I am on the remediation and chemical side of conservation, but we go hand in hand. I'm really interested. But there isn't a lot of work currently being done on that, at least not anything that's like within the public eye at this time. And funding for projects like that is harder to get than usual right now. So I do think that when the tide shifts on funding, there will be lots of mycological projects coming in the realm of academic science. Other questions? Yes.
Will you be leaving any mushroom walks around here anytime soon?
The question is, will I be leading mushroom walks around here anytime soon? I don't think anytime too soon. I have a few conferences in September. That's mushroom conference season. And then in October, as the season starts winding down, we'll have a few community events a little closer to Philly. I think we've got one more foray at Evansburg and maybe one at Fort Washington, but not too many out this way on the calendar for 2025. We will be back in the spring.
When you mention your calendar, is that something that people could find on the website?
Absolutely. You can find our calendar at phillymycoclub.com. You'll see all sorts of events on there. I've got 5 next week. And last year my club did 112 events throughout the year. So we're all volunteer run. So we're doing them constantly. And we try and get out as far as we can with anybody who asks. So if you put in a request, we'll try and make it happen.
Well, thank you very much.
Thanks.
Thank you for coming out and sign up for the Philly Myco Club and become a member of French and Pickering and do all kinds of good in the world. Thanks for coming out.